Hi everyone, my name is Callum. I'm in the third year of a PhD. We're in the University of Manchester in the UK, and today I'm going to be talking to you about how in situ phosphate mineralisation could be applied to the Sellafield subsurface in the remediation of radioactively contaminated land. So a bit of an agenda for today's talk. I'm going to very briefly go through a bit of background about Sellafield, as I'm sure you heard a lot about during yesterday's session, and then discuss why phosphate mineralisation might be a really good approach to remediating a variety of different radionuclides at Sellafield, and then talk you through some experimental systems and the results from these systems that we've been looking at recently, and how these could directly impact the Sellafield subsurface remediation. So just very briefly, a bit of background about Sellafield. So the site was first constructed during the 50s for the UK's atomic weapons programme as a plutonium production facility. And in the later years preceding that, it was used mainly as a reprocessing facility for spent nuclear fuel, originally for the Magnox fleet of reactors and then subsequently for the advanced gas core reactors in the UK. A number of these buildings and silos, the picture on which you can see there, originate from the 50s when the site was first constructed. And these have unfortunately led to the contamination of the subsurface beneath the site, beneath where these buildings are, which has led to radioactively contaminated land and radioactively contaminated groundwater plumes migrating beneath the site. There are a number of different radionuclides within these plumes, but I'm mainly focused on strontium-90, and that's because strontium is very poorly attenuated by the Sellafield subsurface in comparison to other radionuclides within these plumes. This presents a risk that these migration of these plumes could eventually end up pushing the strontium further away from its source of contamination. So my project is mainly focused on how to remediate these strontium-90 contaminated plumes, predominantly focusing around in situ phosphate mineralisation techniques. So one question you might ask yourself is, why bother with in situ mineralisation? Well, one thing that this picture nicely demonstrates on the left is how congested the Sellafield estate is. A number of these buildings, well, quite a lot of these buildings were actually built when the site was actually opened, so there's a lot of ageing buildings and a lot of high level of congestion on the site. So it doesn't lend itself to conventional remediation techniques such as dig and dispose or pump and treat, due to the lack of available space for infrastructure such as treatment works, and also the issues associated with moving large amounts of contaminated subsurface and excavation next to these legacy facilities. So this is where phosphate mineralisation could really, and other in situ treatment techniques could really benefit the Sellafield remediation. Phosphate mineralisation techniques rely on the formation of insoluble calcium phosphate phases such as hydroxyapatite. These phases have the ability to incorporate a variety of different radionuclides such as strontium-90 within their structure. The strontium in the environment behaves very similarly to calcium, them both being divalent cations. You can get significant ion exchange of strontium with calcium in the hydroxyapatite lattice. Once formed, these apatite phases are very recalcitrant to dissolution because they're so insoluble in environmental conditions, which makes them really good radionuclide sinks. One question you might be asking yourself is, why don't we just inject phosphate containing solutions into the subsurface to promote the formation of these phases if they're so great at removing radionuclides? Well, one issue with that is because they're so insoluble, they drop out of solution pretty much instantaneously the second they see any calcium. So if you just inject a phosphorus-rich solution down a borehole or an injection well, you just lead to the formation of a phosphate-rich rind at the bottom of the borehole, which prevents the widespread distribution of the amendment solution into the subsurface. You only see a relatively small treatment area, and it can lead to the blocking of these wells through the formation of these phosphate minerals. So one area of remediation that's seen quite a lot of interest over the years is how we can use different amendment techniques to form these phosphate minerals slowly, which enables a nice widespread distribution of the mineral and the amendment solution in the subsurface, increasing your targeted treatment area and preventing the blocking and clogging of these injection wells. So look, most of the work in the past has focused on these three different systems here. I'll very briefly go through how these all work. So firstly, I'm going to talk through the calcium citrate sodium phosphate system. So this is a biotic process that relies on the microbial degradation of a calcium citrate complex that you inject into the subsurface alongside a phosphate containing solution. So the calcium citrate, because it's complex together initially, you prevent the rapid formation of calcium phosphate phases. As I alluded to earlier, that's beneficial because it means you can increase the distribution of your treatment zone. But over time, the citrate molecule is degraded by microbial processes in the subsurface using the indigenous microbial communities. This then releases aqueous calcium into solution, which over time will react with the aqueous phosphorus that you injected alongside and form calcium phosphate phases such as hydroxyapatite, which will then pull out your strontium and your other radionuclides from your contamination plume into these really favourable radionuclide sinks, these calcium phosphate phases. And as alluded to earlier, this has been looked at really well and deployed at full scale at the Hanford nuclear site in remediation of one of their strontium-90 plumes, but has yet to be looked at in the UK. Really. So glycerol phosphate, again, this is a biotic process, but here instead of controlling the calcium concentration in the groundwater, we're controlling the phosphate concentration. So we're relying on microbial degradation of glycerol phosphate. So phosphates, because they're such a ubiquitous mineral in many different microbial processes, many different microbes have developed an enzyme to cleave them from organic containing molecules. And the phosphatase activity is ubiquitous in many different species. So once you inject glycerol phosphate into the subsurface, the indigenous microbial community will cleave the phosphate, liberating it into solution, which can go on to react with calcium present in your groundwater, form these calcium phosphate phases, which will then hopefully pull out the strontium alongside. And we've looked at this quite heavily in my group, in our group in Manchester, with respect to uranium and strontium. And then finally, polyphosphates. These are a little bit different. These rely upon an abiotic process, essentially just the hydrolysis of long chain polyphosphate. So lots of orthophosphate units stuck together, and the longer the chain, the longer the hydrolysis that it takes. So you can slow down the release of aqueous phosphate into solution by combining lots of phosphate molecules together. Again, you see, hopefully you'll see the same formation of these phases and the pulling out of strontium. And this has been looked at Hanford before, but it was predominantly to do with uranium sequestration rather than strontium. So my project's looking at essentially, will these three different amendment solutions work in a Sellafield type situation? So I'm very briefly going to go over some experimental results that we have recently concluded. And firstly, it's worth me talking about the systems that we've looked at. So I'm only going to talk about the calcium citrate and sodium phosphate and glycerol phosphate systems today. I don't really have time to talk about the polyphosphate one as well, sadly. And essentially what we do is we try and simulate Sellafield in a bottle. So we use representative groundwater and representative sediments from the subsurface at Sellafield, which is actually quite a heterogeneous system. There's various different sediment types beneath Sellafield, ranging from really lean, sandy material to much more iron oxide and clay rich. So we have run these experiments in parallel with much more clay on our iron oxide rich material. But I'll just be presenting this kind of lean, sandy material system here. So I'll go into some aqueous data now and the results from these experiments. So the calcium citrate sodium phosphate system worked really well at remediating strontium from the aqueous phase compared to just adsorption to sediment, which is shown by the dotted line. So we see co-removal of strontium, calcium, and phosphorus over the duration of the experiment, suggesting that they were all pulled out together during the same process. And at the end of the 30 days, we took the sediment and ran it under SEM for SEM analysis, and we saw the formation of these spherical nanoparticle agglomerations, which is characteristic of bioprecipitated hydroxyapatite phases. We looked at this using EDS analysis, so point EDS, and saw they contained calcium, phosphate, and strontium within these phases. So we also took the end sediments down to the diamond light source at Harwell in Oxford. This synchrotron was then used to do Zane's and XF analysis on the sediments using the strontium K edge. And these preliminary fits shown here add additional evidence to suggest that the strontium is incorporated into hydroxyapatite rather than any other calcium phosphate phase, as these parameters are consistent with strontium incorporated hydroxyapatite. So moving on to the glycerol phosphate, we see similar trends in the aqueous data. However, the overall effect is slightly less removal in the glycerol phosphate system than in the calcium citrate system. But again, we see the co-removal of the calcium and strontium, suggesting they're being pulled out in the same phase. Now here in this system, we'll actually see the ingrowth of phosphorus, because we start with none, but as the organophosphate is the organophosphate molecule is degraded and the phosphate is liberated microbially, we see an ingrowth in phosphate, reaching a kind of equilibrium at around 20 days. Again, we took the endpoints of this experiment for SEM analysis and saw some nice spherical agglomerations of these nanoparticles together, and the evidence of phosphorus, calcium and strontium within these from EDS analysis. So we also performed XAS analysis on the endpoints from the glycerol phosphate experiment using the strontium K edge, and here the fitting parameters shown below are consistent with strontium incorporated hydroxyapatite, and these preliminary fits seem to be consistent with the SEM imaging, and indeed is consistent with the fitting parameters used in the calcium citrate system, suggesting that both systems have formed the same product. So to conclude, we've hopefully managed to convince you here and show you that both the calcium citrate and glycerol phosphate systems work remarkably well at removing strontium when compared to just the basic adsorption to sediment, and this has implications as they could be applied to the Sellafield subsurface in the remediation of strontium-90. We've shown that the phase formed is strontium incorporated hydroxyapatite, the most favourable phase in these systems for removing the strontium and for keeping hold of the strontium long term. And then the next steps really in these experiments are to make it as close to the real site as possible. So we're going to do some flowing column studies shortly, and hopefully with some active strontium-90 as well, so we can get down to proper environmental concentrations and look at whether strontium-90 behaves in a similar way. So thank you very much for listening. I'll be happy to take any questions you have.